So far we have looked at emotions from the point of view of psychology or philosophy or in arts, but ultimately since this course is about brain, we should talk about emotions as occur in the brain. What is the neural basis of emotions? How does brain process emotions? So, in neuroscience very often if you want to study how what is the contribution of a given part of the brain to a given aspect of behavior or experience, right. These studies come from say pathological situations where something has gone wrong with that part of the brain. So, we have seen that if the temporal lobe is damaged or hippocampus is damaged, the individuals have problem with memory storage. So, we know that that part is involved in memory storage, right. So, similarly in case of emotions also, certain experiments have shown that certain parts of brain contribute to emotional experience. In some of the earliest experiments of this kind, which are slightly crude, so they have actually taken cats in which the cortex is removed. So, you have what is called a decorticated animal preparation. So, they can remove the entire cortex, the surface of the brain. When they did that, the cats exhibited signs of emotional arousal. And so, they would very easily get provoked, right. They would crouch down, arch their backs, retract their ears, they would growl and hiss, right, all the, and that this is all external expression. But in addition, they would also show the full autonomic arousal, the activation of sympathetic nervous system, like piloerection and pupil dilation, a higher blood pressure and heart rate and so on and so forth. So, once the cortex is removed, it is as if something is, some inhibition is removed, and the animal is easily provoked into a rage and expresses a full blown sympathetic activation. And on the other hand, lesion to hypothalamus, we have seen hypothalamus before, we will also be visiting it very soon, right. Lesion to hypothalamus highly attenuates emotional reaction. So, there are parts of the brain which when damaged, right, have influence emotion in some form or other. So, here is a picture of an animal showing all this hissing and an aroused behavior, and the picture you also see on the right the location of hypothalamus in the brain. So, we have seen earlier that hypothalamus lies ventral to thalamus and has extension connection to thalamus and midbrain, and also it is it's a kind of it controls the entire the endocrine system, the system of glands. It also receives and controls information from autonomic nervous system. So, it controls the pituitary gland, which is called the master gland of endocrine system, and controls its hormonal secretion. It controls body temperature, emotions, hunger and thirst, and circadian rhythms. Now, there are certain areas in hypothalamus which when stimulated, right, produce different kinds of very strong emotions. So, for example, there are some areas which when you stimulate causes pleasure. So, some of the earliest experiments, right, with animals where they inserted electrodes in certain locations in hypothalamus, right, the animal preferred to stimulate itself, right, by pressing a lever, right, and at the cost of even foregoing food and starving itself to death. Okay, so, these are the pleasure centers of the brain. The other centers in hypothalamus where they are electrically stimulated produce rage or fear. Then other places when stimulated give inhibition of pain. So, the hypothalamus is a very, although it is a very small nucleus, it has lot of centers which when activated have some kind of emotional expression. Then there is this syndrome called Kluver-Bucy syndrome, right. So, basically in this syndrome is exhibited by animals in whom there is the temporal lobe is lesioned on both sides. These animals show no fear or aggression, they become very tame, they show exaggerated oral behavior. So, kind of they try to put everything that they see in their mouth. Oral behavior is also seen in infants, right, you see babies putting everything in their mouth, and they also tend to react and overreact to every visual stimulus. They also tend to copulate in a kind of abnormal fashion with any object, right, or with animals of other species. Okay, so, so again you can see that damage to certain part of the brain can give rise to certain abnormal emotional behavior. There is another organ which is also in the temporal lobe, right, a brain structure in the temporal lobe called amygdala, right, and if you pair a neutral stimulus with a painful stimulus or an aversive stimulus, and the response to the neutral stimulus can be learned. Let us look at an example. Here in this picture you have a rat in a cage. So, in the experiment they play a sound which is neutral, emotional neutral, and then after the sound after a little delay, a current is passed in the kind of a grid which is at the bottom of the cage, and because of the current the animal receives a shock in its feet. So, the shock is pretty strong, therefore, the animal has a panic reaction and kind of it freezes in its current position as a way of expressing its fear. So, in the experiment what they do is they repeatedly pair the sound with the shock, and after it is done for a sufficient number of trials, then when the sound alone is played, the animal again shows panic reaction as if it is also receiving the foot shock, although there is no actual foot shock. So, this is a kind of example of a classical conditioning where there is a pairing of a neutral stimulus with an aversive stimulus. What is interesting is this pairing does not occur if the amygdala is lesioned. So, which means that amygdala is necessary for learning this expression of fear, for learning to bind this neutral stimulus with the aversive stimulus, which is in this case the foot shock. So, also amygdala is also has an involvement in aggression, for example, when it is overactivated that can give rise to increased aggression. So, one example is there is this case of a person called Charles Whitman, who went on top of this university tower in University of Texas in 1966 and started shooting blindly. A lot of people died in that accident. When this individual's brain was studied on post-mortem, it was found that his amygdala, there is a tumor pressing on his amygdala. So, that therefore, it was getting overactivated, and that gave rise to aggression. Therefore, underactivation or simply lesioning of amygdala gives rise to reduced aggression or placidity, which is what you see in Kluver-Bucy syndrome. Okay, so, you can clearly see a correlation between amygdala and aggression or fear, right, or fear at the other side of it in these studies. So, lesion leads to Kluver-Bucy syndrome, right, and the lesion of temporal lobe can give rise to lesion of amygdala, right, and then because of this animals become tame, they show no fear or aggression. So, in this picture you can see amygdala, and which is right next to hippocampus, both are inside the temporal lobe. We have already seen hippocampus before in the context of memory, but hippocampus also has some connection to emotions. Hippocampus we know is involved in memory consolidation and spatial navigation, but it is also involved in fear conditioning. So, for example, the fear conditioning by cue, which is in this case a tone, right, is one kind of conditioning, but the conditioning can also be done by the context. For example, in this cage that we have just seen, right, the animal is in the cage, and when you play the sound, when sound is paired with a foot shock, right, next time when you play the sound alone, the animal shows fear reaction or panic reaction. But suppose the animal is in certain cage when this kind of a pairing occurred, and next time around when you bring back to the same cage, immediately it can show a fear reaction to the cage, because in this case the animal has learned to pair its context, its surroundings with the painful stimulus, which is the foot shock. So, there are two kinds of pairing that happens in these experiments: the pairing of the tone or the cue with the aversive stimulus, and the pairing of the context, which is the cage itself, with the aversive stimulus. So, it turns out that on amygdala removal, the fear conditioning to both cue and the context was eliminated, but on removal of hippocampus, fear conditioning only to the context was eliminated. So, hippocampus has a role and fear conditioning to only the context. So, we have seen several brain structures, all of which contribute to emotion experience in some form or other. So, when people have studied the different parts of the brain which have a particular influence on emotion, they put all these studies together and have created this idea of a limbic system, right, which is the subcortical network which forms our emotional system. So, there are all these components, right, all these brain structures within the subcortex, like hypothalamus, amygdala, hippocampus, mammillary bodies, and cingulate gyrus, which is actually a part of the cortex. All these brain regions form a network which is called limbic system, and this is the emotional part of the brain. Okay, in this picture you can see certain components of the limbic system. So, you see thalamus here, then hippocampus is here, amygdala is here, and you have mammillary bodies also here, the hypothalamus is here, and the cingulate gyrus, right, is this big region is cingulate gyrus. All these areas form a network which is the limbic system and which is the emotional machinery of the brain. Now, let us look at two fundamental axes of emotions. So, there is fear, which is processed by amygdala and hippocampus, and there is pleasure, which is processed by basal ganglia, which is a whole different circuit. Okay, you can see in this picture basal ganglia, which is also a subcortical circuit, which consists of several nuclei. You can see some of them here in this picture. So, for example, there is the caudate and putamen, and there is substantia nigra, globus pallidus with its two components, the externa and the interna, and the subthalamic nucleus, right. So, the basal ganglia receives input from the cortex and sends output back to the cortex, and this circuit has actually two pathways in it. So, the input from the cortex comes to one basal ganglia structure called a striatum, which is this whole region, and the output from striatum goes directly to GPA and onwards back to the cortex by a pathway which is called the direct pathway. Then the output from another output from striatum goes to GPE, subthalamic nucleus, STN, and then GPI over a longer route which is called indirect pathway. So, then there is this substantia nigra, which releases a substance called dopamine, which acts on striatum and controls the flow of information from cortex and through the basal ganglia. Now, this dopamine is a very important chemical which coordinates the activity of various nuclei in basal ganglia. Dopamine is called the brain's pleasure chemical because it is released when brain is stimulated. So, for example, we talked about the pleasure centers in hypothalamus, right. So, similarly, the other pleasure centers in, for example, nucleus accumbens, if you stimulate any of these pleasure centers electrically, right, that causes release of dopamine from substantia nigra. Or if you take drugs or opiates, right, or even food, because food can be pleasurable. So, these kinds of stimuli cause release of dopamine from the dopamine centers. Then dopamine antagonists, that is chemicals which counteract the effect of dopamine, can attenuate the pleasurable experience. So, if you take say if you eat say nice as like a sweet, for example, which would normally release dopamine and gives you a pleasurable experience, if you simultaneously take a dopamine antagonist, then your experience is somewhat attenuated, it is not so much fun anymore. So, similarly, people have studied and shown that the presentation of beautiful faces, right, activates the same parts of the brain which are activated when dopamine is released, or images of lovers. So, couples were made to participate in these studies and they were shown pictures of each other and their brains were scanned in fMRI, and they were shown that the same reward system, right, is activated because of by appearance of the lovers, right, of the spouse of a given individual. Or even presentation of monetary rewards, you give somebody cash, right, you can see the reward system in the brain getting activated. So, reward system is basically brain's an underlying machinery for processing pleasure or positive stimuli. Okay, so, this machinery can be used for learning, right, because very often when you learn something, you are often learning a mapping between a stimulus and a response, and if your response to the stimulus gives a positive reward, right, then next time around you tend to repeat that response to that stimulus, right, so which is called reinforcement or learning by reinforcement. Or for a given stimulus, if a response gives rise to punishment, then next time around you try not to repeat that response for that stimulus. Okay, so this kind of a learning by reward and punishment is called reinforcement, and basal ganglia plays a major role in this form of learning. And that is because if you look at the anatomical connectivity of basal ganglia, it is located very strategically in the brain. It receives the input from the sensory and motor cortex. So, in this picture you can see that, right, there is sensory cortex and motor cortex, right, you receive input from the world in your sensory cortex, and which sends information to the motor cortex sends commands which will make you move and act on the world. So, this information is sent to basal ganglia, and basal ganglia also receives information from the substantia nigra, which sends information about the reward. So, in this figure you can see that, so there is this box, right, which combines three forms of information: the stimulus, the response, and the reward. So, if you look at what basal ganglia is doing, it has information from the cortex about the stimulus and the response. It has information from substantia nigra about the reward. So, it is combining all these three and sending it back to the motor cortex and giving a recommendation about what is a good action to perform. So, there are also reward sensing neurons in prefrontal. So, in addition to basal ganglia, there are other parts of the brain which also process reward or pleasurable experience. So, in this experiment the subject is shown a flower or which is a stimulus. In response to that, it is expected to pull a lever or perform an action. When that action is performed, it is given a reward like juice. Okay. And then there is another stimulus which is another flower, a yellow flower, and in response to that it has to hold if it holds a lever, then it is given a neutral outcome like the sound. So, when they presented this and the animal is trained on this kind of a task repeatedly, okay, what was found is in early stages it only knows about the reward, right, and at late stages the neurons were able to learn in response to reward and the intended action. So, in the early stage neurons respond to the anticipation of reward, right, that it is going to get a juice or something like that, but after a delay neurons firing had represented both the anticipated reward and the intended action. So, basically the animal is the neural firing indicates that the animal thinks that if it performs this action then it is followed by a reward, right, because it is seeing this kind of a particular stimuli. Okay, these kinds of neurons were found in prefrontal cortex, right, particularly in the cingulate sulcus. Okay, let us look at one more element of neurobiology which has something to which will finally link to our outline of theory of emotions which has its roots in neurobiology. So, there are these neurons called mirror neurons, right, which fire when animal acts or performs an action or when it sees another animal, right, performing the same action. Okay, so, it fires when both animal acts and when animal observes the same action performed by another animal. So, for example, right, this mirror neuron responds when a monkey rips a piece of paper, right, and the same neuron will respond when a monkey sees a person ripping a piece of paper or even when it hears, right, or auditorily, right, the sound of paper getting ripped. Okay, that means the mirror neurons encode abstract concepts of actions like ripping paper. The action could be performed by the animal itself or some other animal. Now, it has been thought, so since the neurons respond the same way, this mirror neurons respond the same way, whether the action is performed by yourself or somebody else, people propose that this system, this mirror neuron system is necessary for imitation learning. And imitation learning is found in very small animals. For example, you see in this picture a person is sticking his tongue out and the little monkey cub, right, is able to imitate him by sticking its tongue out. So, mirror neurons, so it is not just a motor function which mirror neurons reflect, they also are found to reflect emotion. For example, there are certain brain regions like anterior insula and anterior cingulate cortex and inferior frontal cortex. They are active when people experience an emotion like disgust, happiness, etc. And they are also active when they see somebody else experiencing the same emotion. So, which means there is a mirror neuron system for emotions, and it turns out that mirror neuron dysfunction happens in autistic children, and therefore, they have trouble understanding emotions in others, and not only that, some autistic children are thought to have the problem of theory of mind. They do not seem to have what is called theory of mind, that is the understanding that just as they have a self or a sense of self, others also have a sense of self. So, some autistic children seem to have trouble with having a theory of mind, because there is a problem with their mirror neuron system. Again, there are reward neurons which distinguish between one's own and others' rewards. So, whether you get a reward, right, or somebody else gets a reward, there should be brain should be able to distinguish between these two. Otherwise, you cannot really operate in the real world, because if somebody else gets a reward and if you think you have got a reward, then you will get into troubles of decision making. So, similarly, people found differential activation of the brain during social competition. So, when you are competing with somebody, when somebody else is winning, the brain should respond to it in one way, and when you are winning, you should respond differently, otherwise you will not be able to compete. So, brain actually shows that kind of a differential activation. So, similarly, when somebody else performs an error as opposed to you performing an error, right, different brain areas are distinguished between these two cases. So, to summarize some of the ideas that we have looked at. So, we reviewed some of the observations from neuroscience, right, which have relevance to emotion processing. So, main ideas are that basal ganglia process pleasure and amygdala processes pain or aversive stimuli, which gives rise to fear. Then the other idea from neuroscience which we invoked is the mirror neuron system, which can distinguish between something happening to me and something happening to other. So, with this we will give a quick outline of a neurobiological theory of emotion, but let me say straight away that we still do not have actually a comprehensive theory of emotion. So, I think here in the lecture we are trying to put together ideas from many sources and trying to give a kind of a cartoon picture of how emotions work in the brain. So, there is this interesting paper by Beeman et al., right, where they have tried to reduce a lot of emotions to certain fundamental dimensions, right. For example, happiness they define as being pleased about a desirable event, okay, and unhappy is defined as displeased about an undesirable event, or happy for is defined as pleased about an event desirable for another, or if you go down the list, right, pity is displeased about an event undesirable for another. So, something that is undesirable for somebody else has happened and I am not happy about it, and that is what is called pity. Or similarly, if you look at some of the other examples, fear is being displeased about a prospective undesirable event. So, something undesirable is going to happen and I am not happy about it. Or if you look at shame, disapproving of one's own blameworthy action, or pride is approving of one's own praiseworthy action, and so on and so forth. So, this way of defining emotions basically is reducing emotions to certain basic dimensions. So, most of the previous list of emotions can be expressed as response something good or bad that has already happened or is currently happening or going to happen, right, in prospectively to oneself, or all these things to somebody else. Okay, so, if you look at all that we have covered in terms of neurobiological substrates of emotion, we have seen that the brain areas which tell you whether something good or bad is happening, right, and the brain areas which tell you whether something is happening to you or to somebody else. And I have not covered in this lecture, but there are also brain areas which or brain signals which can tell you about what is going to happen in future as opposed to what has already happened in the past. So, if you combine these ideas, you kind of see an outline of neurobiological theory of emotion. So, now let us make a summary of what we have discussed so far in this lecture. So, we have presented we have begun the discussion with philosophical theories of emotion. We discussed ideas from both Indian philosophy and Western philosophy. Then we talked about classification of emotions based on facial expressions, we are starting with Darwin and others like Sylvan, Paul Ekman and others. So, then we talked about psychological theories of emotion, right, the James Lange theory and Cannon-Bard theory and so on. Then we talked about neurobiological basis of emotion, like that positive evaluations are done by basal ganglia, which is pleasure. Negative evaluations, that is pain or fear, is done by amygdala, and finally, the distinction between self and other is done by the mirror neuron system. So, finally, we have put all these ideas together and proposed a kind of a neurobiological theory of emotions. Like again, let me repeat that we still do not have a comprehensive theory of emotions, but I think there are a lot of pieces of necessary ideas to build this kind of a theory. These ideas can be borrowed from psychology and neuroscience and so on, right, and this is all kind of something that is left for future research. Thank you.